speaker of the afternoon, final speaker of the entire workshop is George Stetten. George is here at Carnegie Mellon and is, he has joint appointment with CMU and the University of Pittsburgh. Listed here as Carnegie Mellon.
That's because we're at Carnegie Mellon.
Yeah, I guess so. His title is In-Situ, Virtual Images and Virtual Forces to Augment Image-Guided Intervention.
Equations than the previous one, and pretty much all of the others. In situ virtual images and in situ virtual forces. You haven't heard about virtual forces before, probably. And I'm going to try to make the case that the two things that I've been working on mainly for the last 10 years are actually variations of the same thing. And we'll see if it works. I'm at Pitt and at CMU, and I'm also at CMU, I'm at the Robotics Institute mainly. So this is not fundamental ultrasound. This is display and haptics using ultrasound and other imaging modalities. And the main thing that I have built my last 10 years of my career on was a very simple idea of taking an ultrasound probe and looking at the image that it creates with a little display that's mounted right on the probe with a half-silvered mirror, so that when you look through the half-silvered mirror, the image that is being displayed is reflected as a virtual image inside the body in its actual location, so that you can actually stick a needle into the image. It's floating inside the patient at its actual location, so that the ultrasound technician or the clinician doesn't have to look away from the patient at a screen. The image is actually where it should be, where the data is, because virtual images are stable if they're created with a mirror. You move your head around, they stay where they are, and your two eyes focus on it, and convergence and stereo vision all works appropriately, so that you really do think that that image is there. It's as if the photons were coming from inside the patient. So we built our first one without any funding from Home Depot, with a little display and a ultrasound machine and a big half-silvered mirror. And you'd look down through the half-silvered mirror, and sure enough, there was the ultrasound scan floating at the end of the transducer, and it looks sort of like a flashlight, so I called it the ultrasound flashlight, but that's too long, so it became the sonic flashlight, which is now a trademark owned by the University of Pittsburgh. So you're looking down through the mirror, and you put your hand up against the ultrasound probe, and there's the inside of your hand as an ultrasound image in real time, floating right, not on the surface of your skin, but actually inside your hand, where it actually is. If we could, some of my pictures are a little bit hard to see in the light. It might be good. So when you move your hand around, yeah. Oh, okay. Oh, it's one of these. Yeah. All right. Good luck. Oh, wow. That's volume.
Volume is not the same.
Yeah, down. Everything down. Chandeliers, maybe. Ah, that's better. Okay. So let me run that again. So there's your hand, and as you move your hand, the image stays where it is, but your hand is moving and the bones are moving. And so it's calibrated once at the factory, basically, and there's no tracking required and no head-mounted display, and all of the other more complicated methods that people have used to do what they call augmented reality, putting medical images in the visual field of the doctor, which is obviously the original form of medical imaging is the visual field of the doctor, and it should be merged with all the other medical imaging modalities. Is done optically here rather than with tracking and head-mounted displays, virtual reality techniques. So it's a lot simpler. We got an inordinate amount of press. We were in Discover magazine. We were on the front page of the Scientific American. And they started using words like flesh rather than tissue, as the press gets involved. This is way back in 2002 or so. We're not the first to attempt to place medical images within the surgeon's direct line of sight, but it's the first to succeed without the patient wearing any sort of tracking device or the doctor wearing some sort of a fancy goggle system. And we did a lot of putting balloons and poking fingers into balloons. And it gives you an eerie feeling to see the ultrasound image at your fingertip and have it actually move around with your finger. I can't really do it justice, but when you see it and it's stereo and it's live and it's actually floating in 3D, you sort of feel like Superman. You're looking through things. And then we put a tube in a balloon, and we knew that we were going to be talking about palpating vessels and sticking needles in vessels and stuff like that. And you could stabilize the vessel, and it really is a kind of a cool feeling to see the ultrasound right on your fingertip moving around with your fingertip. J&J came and wanted to see us stick an olive in a turkey breast. That was apparently their gold standard. So we stuck an olive in a turkey breast, and there it is showing up. And this was the first biopsy using the sonic flashlight. You stick the needle, it was so clear, there it is. I mean, you're looking through a mirror, so there's this, what we call the sneeze guard effect. You have to work under this piece of glass like you do when you go to the cafeteria, but you get used to it. And it was clear where the olive was, and sure enough, when you stuck the needle in, you hit the olive. We started making it smaller, so that we could move it around like an ultrasound machine. We made one that had 27 degrees of freedom and was never calibrated correctly. And then we made one that wasn't adjustable and was calibrated. And we worked our way toward getting smaller and smaller. And so this is our little portable, somewhat portable version. And again, it had this nice response to the tool showing up in your visual field. Your hands are in the visual field. The patient is in the visual field, the ultrasound is, and it's all the same. It's all fused into one 3D reality. And you're very used to that because since you've been a baby, you've picked up tools and held them in front of yourself, and you know where the end of a tool is. Even when it disappears, when you're drilling, the drill bit, you know where the drill bit is just intuitively. You don't have to see it. If there's a shaft and it disappears, you know where the other end of it is. We kept on getting press. So they grabbed this off my website in Popular Mechanics and showed it being used at the scene of an accident. And we hadn't even had animal protocols yet. So, we started getting calls from fire departments that wanted to buy one. We went to cadavers. This is an eye being scanned with our little portable device. And it was interesting because we had designed it to be held a certain way, and the doctor picked it up and immediately held it the wrong way, which turned out to be the right way. So we work closely with a lot of doctors on this. There's a patient. We're scanning the eye. There's the eye inside the patient, and there's the display. And when you look through it, you see, it's a little hard to make sense of this, but there's the eyeball, there's the optic nerve, and it's floating inside the patient. And you can stick a needle behind the eye without hitting the optic nerve. We got an NIH grant eventually and started building a serious one that could be used in patients. It does have to be calibrated, so here's as much math as I'm going to have. Angle of incidence equals the angle of reflectance. It's nice. It's sort of MD math. I'm an MD. And so there's the ultrasound probe, and there's the image, and there's the display. There's the mirror. You basically bisect the angle. And you do have to calibrate it so that this display has 6 degrees of freedom if it's got the right scale, 7 if it's got isometric scale. So it's an affine transform, and you solve it once. We have a little phantom. This was a graduate student, Wilson Chang, who's now chief resident of radiology at Stanford. And he proposed a gel that was very thin that would have little beads in it that you could see without any refraction. A water tank doesn't work because you got to actually see the target, so you need a very thin gel. I didn't think it would work, but sure enough, this is just a commercial ultrasound probe with the display on it. Sure enough, the ultrasound goes in and out of that gel just fine, and we can make the little dots appear in the ultrasound image at the same place as we can see them. And if you have 3 of them, then you can solve the affine transform and get the thing calibrated. So this was the clinical machine. This is a tube in a phantom. And anybody comes into my lab, I still have this. It's a very reliable phantom, a very reliable demo. Anybody comes in, sees the tube, picks up the needle, and hits the tube on the first time, whether or not they've ever used ultrasound or stuck a needle in anything. We talked to our clinicians, and they said that PICC line placements would be the first use that we we could probably benefit with this. PICC lines are central catheters that go in through the deep veins in the arm into the heart, and they become the bread and butter of radiology. They don't even want to do them because they have so many of them. So nurses are now doing them with ultrasound guidance, but they have to look away. And if they could look right into the arm, they do it at the bedside. So that became our clinical trial. Before that, we did some studies that showed that people, untrained people as well as nurses, could learn to stick the needle into the tube faster with the sonic flashlight than with conventional ultrasound. And we showed that they had a higher success rate. And then we went on and got an IRB and did a clinical trial. There's one of the veins that we would go after. It's I forget the names of the vein, the brachial vein, I think. Actually, I forget. I should know that, but medical school is fading. But the nurses did it. There's a vein and an artery and two arteries in a vein, I guess, and the nurses really liked it, and it worked, and we did a successful first clinical trial with the device. This is what it looks like on a cadaver. There's the vein, and there's the needle going in, and you can see that the needle lines right up. And once you've calibrated the device, it's calibrated forever. So that's basically the sonic flashlight and its first clinical trials. There was problems with sterilization because the ultrasound probe needs to be inside a bag, and then the mirror was inside the bag, and that didn't work, so we had to spend a whole year figuring out how to get the mirror outside the bag, which we came up with a solution, which I'll show you in a minute. Other possible clinical uses, this is a big one. There's the jugular vein, and the carotid artery is the big one, and the jugular vein is the little one. You can tell it's the vein because when you press on it, it squishes. And these are often cannulated in the middle of the night by a first-year intern, and untold numbers of people are actually killed by it, and they don't even have good statistics because the patients are often dying anyway by the time they get to putting a catheter in an emergency situation. But if they could make sure that they don't hit the carotid artery, which is a bad thing to hit because that sends a clot into the brain sometimes, and hit the jugular. So this was proposed. We haven't actually done a clinical trial with this one yet. But that's what it would look like. There's the jugular vein with a needle in it. This is a cadaver. Other possibilities, this is a joint, sticking a needle into a joint to deliver drugs or to take a sample. You can, the bones are black, and you can actually make a good ultrasound. It's pretty useful around joints. This is a thyroid biopsy. As the needle goes in and out, you see the image going in and out. It's a little bit of lag, because in this early model we had a little bit of lag in our processing, but nowadays the graphics is so fast that there's no delay anymore. This is brain surgery. There's a brain surgeon with a cadaver with a head, just literally a decapitated head, and he put a balloon in there, and he could see the balloon in situ. This was an instant article in Neurosurgery with a note from the editor-in-chief saying that this was going to facilitate the use of ultrasound in neurosurgery, because the neurosurgeons, they really can't afford to look away at an ultrasound scan. And so they don't use ultrasound as much as they might, but if the image was floating right on the surface, inside the surface of the brain, they might use it. So when we started to get some interesting things here, we were on the cover of Wired magazine, X-ray vision. It's not X-ray, of course. And they said it would let you see through skirts, which I thought was an interesting application. I had never thought of that before. That was Wired's contribution to the research. We started thinking about commercialization. We figured that you could put gears on it and it could fold up like a cell phone, because ultrasound is eventually going to be in everybody's pocket. It's going to be like, don't use my ultrasound machine, get your own, They're going to be the size of iPhones, and they might flip open. In fact, here's our early model that's also a cell phone. You can take calls on it. This is the commercial model. We have a company now in Pittsburgh called In-Situ View, In-Situ View, if you will. I'm on the scientific advisory board, but I don't have much to do with them because the University of Pittsburgh has a lot of conflict of interest situations. But there's a little disposable, and then the company loves a disposable because then they can, make money off of the batteries rather than the flashlight. And the bag goes over the sonic flashlight, and then the little thing clips on with the mirror. And hopefully this is going to be out as a product by the end of the year, but they've been saying that for couple of years now, so we'll see if it actually happens. At the same time as I was developing this at Hopkins, a couple of folks were developing the same idea on CT. This is Russ Taylor's group. Ken Masamune from Japan and Gabor Fitzinger had a CT version of the same idea. And it was independently invented by both of us. Masamune came up with the concept, basically. And it's still an ongoing project. Ken has since gone on and built a MR version of the device in Japan. We went on to try to, do variations on a theme. We built an extremely small sonic flashlight. We call it the sonic pen light, using actually, this is an intercardiac phased array catheter that could create an image, and so you could see pediatric vessels and small things. We built one based on the 3D ultrasound scanner from Duke University. I spent my postdoc working with Olaf von Rahm, helping to develop the phased array, the 2D phased array at Duke. So we actually had his prototype shipped up here, and we built this was my PhD, was finding the heart beating in the 3D data. So I was familiar with that device. And what's neat about the 3D machine is that it can create these C-mode slices that are en face rather than B-mode, which is sort of going out away from you. En face is more like an actual flashlight. It'll actually, it'll be a slice that's like this. So the mirrors and the display are oriented differently on the 3D version with a C-mode slice. And what it looks like is, this is our prototype. It's pretty rough. But you're now looking at an en face image floating at a certain depth within the patient. And that's sort of nice. That's the broadside of a barn. Surgically, that's what you want a flashlight. You don't want to have this thing sort of going away from you at an angle. It's nice to have it facing back at you. And so here's a C-mode image of the inside of the hand. You can see the bones. It's a lower resolution than your regular ultrasound. Especially this prototype was not all that great an ultrasound machine, but it got the point across. We developed a version of the thing which would work with telesurgical devices where the imaging is happening here with a microscope. There's a caviar with a little tiny needle, and you're holding a mock-up of the needle in your hand, and we didn't actually have a robot in this version. We just pushed the egg onto the needle and watched it appear to get impaired on this big blow-up of the thing. So you're holding this mock-up. It's the same angle as the micropipette, and the cell is appearing to be impaled on your device. It's not robotic yet. And then we built a robotic version which had two robots that were master-slave and did the same trick. And so here, the paintbrush is in your hand, but the paint is actually remote on a second robot and a tenth of the scale. So you're painting here. This is David Wong painting his name in Chinese. He's an MD PhD from my lab. And he, the paint is not there, but the brush is. And so it gives you this 3D sense of what you're doing remotely. I won't wait for him to paint his entire name. I've had the benefit of working with a psychophysicist here at CMU named Roberta Klatsky, who's very well known in haptics and vision. And she developed a bunch of experiments that showed that when you're using the sonic flashlight, you actually go for the target because it's actually there, whereas when you look away at a screen, you tend to shoot shallow. And there's several reasons for that. It's interesting. Apparently, this is well known in psychophysics, that when you estimate how big an inch is, you're always a little bit short. People always underestimate how big a certain amount of distance is, if it's an inch or a centimeter. So when you look at the screen and you say, oh, that thing's 2 inches deep, and you shoot for it, you shoot shallow. And also, because of the compression, you have to push the probe down into the patient, so it's actually, off by that distance, and you don't realize it, because that doesn't show up on the screen. You think you're at the surface, but you're actually below it because you're pushing down. So you shoot shallow, whereas both of those disappear when you actually have the image right there. It's in the right place, so you hit it. You also, instead of using homing to get it, you actually just shoot right for it. She did a bunch of really cool experiments. This one shows that when you're hitting targets with the sonic flashlight, you learn how to do it and you learn how to do it, and you transfer that learning to different targets. Whereas if you have a different target with a regular ultrasound screen, you have to relearn how to hit that target because it's in a different location. So the learning transfers better when it's actually just plain old perception. Whereas when you look away at a screen, there's a lot of cognition involved, a lot of higher level thinking that doesn't transfer as easily to different targets. And this just shows that you hit the targets more accurately also. This is a version of the thing, which is another one where the target is actually just floating there in space. It's called grab-a-slice. And it's a way to visualize 3D data, not within the patient, but in a data set that's floating in space. And you actually physically move the screen around through the data set. And this has advantages that we've shown in terms of especially the Z direction, the through plane direction. When you use a mouse to scroll through things, you don't really know how far you've moved in the Z direction. Whereas if you actually move the screen itself, you know how far you've moved. So you can relate X, Y, and Z more directly using grab-a-slice than a conventional display. Another PhD dissertation in my lab. So this may be used for surgical planning or educational purposes, or just interpreting data. What we showed was we used it for pulmonary embolus. We could find whether you were an artery or a vein in the lung by tracing it back to the heart, and you could do a better job of that using grab-a-slice than a mouse or conventional 3D scrolling. So this is all the same theme of in situ, if you will. I'm trying to tie everything we do together. Andrea Novacnik, who is a professor here at CMU before he left, took one look at what I said, and he said, you could replace the mirror with a holographic optical element, which is, in our case, is a clear piece of glass on which a holographic pattern has been built. And if the image is generated using monochromatic light, an LED or a laser, through an LCD shutter, you can create virtual images that are no longer just reflections like a mirror, but can have magnification or can have prisms built into them to move them off the center. So you can basically simulate any optical element in terms of the laser, whereas for plain old vision, you actually are just looking right through it. So it looks like a piece of glass, but the laser image can now be bigger than the source image, or off center or moved around however you want. So we proposed to NSF that this could be used to generate virtual images using laser rangefinders or whatever you have through clouds of smoke for a firefighter. It was right after 2001, and this kind of thing was they were funding, and we got a grant from NSF and eventually from NIH to develop this version of it. And John Galeotti in my lab did his PhD on it, and this is his final product. This is his wife and their first child. This is how he announced the pregnancy to our lab. There's the holographic optical element. There's the image of the ultrasound not being generated in real time, but being projected into his wife's belly. And it's a much bigger image than the transmitter. We can create images that are further away and bigger, which you can't do with a mirror. You know, if you want to see a whole liver, you're going to need a display that's as big as a liver. Which would be a very clumsy ultrasound machine. But the theory was that we could have tiny little transmitters but big images. And halfway through, I just got to tell you, this is the math, the extent of the math. He came to me about halfway through his thesis, and he had found Clark Maxwell, who had said unequivocally that the only stable image, virtual image, comes from a mirror. You can't make a stable virtual image with a lens. It's not possible. And he said, what have you gotten me into? But he then spent the next 2 years optimizing. You can obviously do a pretty good job. And if you, like fancy compound lenses, approximate perfect lenses which don't exist. And so he spent most of his time optimizing it so that the virtual image would only move around a few millimeters at most. And so that's another variation on our theme. The latest one is optical coherence tomography, which is used in the eye to look at the cornea and the retina, mainly the retina, but also the cornea these days. And we're lucky to have one of the great OCT labs in town here. Joel Schumann, the chairman of ophthalmology, was one of the co-inventors of OCT at MIT. And So an OCT scanner is like ultrasound, but it creates a reflective image from the laser light with very high resolution, 3 micron, 1.5 micron resolution is possible. And it can be a 3D data set, very much like ultrasound or a B mode, C mode. They use all the same terms. And that little image, we have a little display and we have a mirror. And under a stereo microscope, you still use a lot of stereo. And you still use convergence, surprisingly, under a microscope. You can adjust the focus of where you are under a surgical microscope using your eyes. So you really do see these little images floating at their actual locations. And we propose to use this to do real-time superposition of OCT data for ophthalmological surgery. And we've built a number of these. And this is our key photo. So there's a needle going into the eye. There's the white of the eye and the cornea. The needle enters the cornea and gets a little distorted by the cornea. There's the OCT image in real time showing you where you are. And there's a lot of corneal surgery that we're proposing to guide using this same principle. Along the same lines, why, if the clinician can merge vision and ultrasound in the clinician's mind, why shouldn't the ultrasound machine itself not be able to see where it's going? So we mounted some video cameras on an ultrasound machine, and they're stereo video cameras, so they can determine where the surface of this phantom is using stereo vision techniques, and can create a 3D world for it to think in, where it knows where the probe is, where the surface is, and where the ultrasound image is relative to itself, so that we can now propose, anyway, to use this thing to give vision to an ultrasound probe. Every device in the world is going to have a camera on it. I mean, there's 100 cameras or whatever, 50, 25 cameras in this room right now. Everybody's got one on their cell phone. Certainly an ultrasound probe should have a camera on it and should be able to see the outside of the patient. You don't expect a clinician to do an ultrasound scan with their eyes closed. Why should the ultrasound probe itself not have vision? You know, it would know where it is. It would know who the patient was. It would know left arm versus right, or what angle it was relative to the skin. It would know how it's moving. We could develop tracking techniques that work in anatomical coordinate systems. We don't need external tracking. We can track the way that a clinician tracks. They look at the patient. They know where they are. So that's another new approach that we're going. It's the same idea. It's the merger of vision and imaging. And then the very last thing is taking this into the domain of force rather than vision. This is the idea of sensation of touch. So this is what we call the handheld force magnifier. It's a handheld tool with a tip that has a force sensor and a solenoid mounted on the back of the hand. And again, the Hopkins Russ Taylor's group has built this, and Tim Salkudian up in Vancouver has built this thing. It's called the Steady Hand Robot or Cooperative Robot. And in their version, instead of having this mounted on your hand, it's a freestanding robot. You grab the tool, the robot's holding the tool. The robot moves around with you and keeps out of your way, and you don't even know it's there. But when there's a little bit of force, it pulls more, so that a little bit of force turns into a lot of force, and you get magnification of the sensation of the force. But it's a rather clumsy device, and all I've done is mount it on the back of the hand so that it becomes a handheld tool. And the force has to be generated from something, so it's generated from the back of your hand rather than from the floor. So, you know, you touch something very delicate, and it actually develops much greater force, so you think it's not as delicate. So you push against this spring, and with the device off, the spring is easy to push, but when you turn it on, all of a sudden the spring seems to be stiffer. And so this membrane in the surgery that is so delicate that you can't feel it, and a lot of microsurgery is done without touch. The microsurgeons can't really feel anything. It's all done visually. But if they could suddenly start feeling stuff using this magnification of force, it might help them out. And we're coming to the conclusion that it's not just microsurgery, that any sharp tool is sharp specifically to minimize force. A needle or a scalpel is meant to cut through like it wasn't there. And so scalpels and needles are inherently hard to feel the force with, but you can have your cake and eat it too, maybe, with just plain old scalpels and needles that are sharp and go in without damaging anything, but give you the force that's more feelable. So it's potentially a broad platform for a new company, and we have a Coulter grant, like a couple of lectures back, there was a Coulter grant. They're to develop commercialization. We have a name for the company. It's called MagnaFeel, like magnify, and we don't have the company yet, but we're looking at forming it soon, and we're testing it on a special device that they have at the Robotics Institute. Ralph Hollis invented this. It's called the magnetically levitated haptic device. It's a 6 degree of freedom device that has huge amounts of force and speed and can know where it is. So we can simulate tissues with it, and we push against it. And we've shown that with the force magnifier, you have a lower threshold of perception of forces, and that you can determine the differences between forces. The slopes of these lines are different. You can discriminate between forces better with the device than without it. Clearly, we need to make it look more medical. We're putting the force sensor inside the handle now, and we're using Boyce coils instead of the big solenoid. This is our current version. It's starting to look a little bit more like a clinical tool. And so this is what I'm claiming now to try to tie it all together. This is my last slide. That the sonic flashlight stuff is direct in situ augmentation of vision to see through the skin by superimposing virtual images using a half-silver mirror or a hologram, and then direct in situ augmentation of touch by superimposing these virtual forces created by the solenoid on the actual forces. And in this case, instead of a half-silver mirror, it's the handle, which adds those two forces together. The half-silver mirror merges the two images, and the handle merges those two forces, so I'm calling it a virtual force. I came up with this specifically for this talk because I had to merge it all together. So I don't know if you buy it or not, but if we combine these two, which we haven't done yet, but in situ images and in situ forces, they may benefit from each other because of the nature of hand-eye coordination in ways that are greater than the sum of their parts. And that's my talk. Hardly an equation for you. And these are the students in my lab and my collaborators and our funding. And if you want to find out more, it's vialab.org. It's our website. We made it. Thank you.
How long, if you gave me a sonic flashlight and told me to put in a PIC line, what's the probability I would get it right on the first time?
Really good.
Like 50-50?
Well, you have to know the anatomy.
Okay.
And there are slight problems because one is that the ultrasound image is much smaller than most ultrasound images. So the detail isn't there. I mean, ultrasound is getting better and better as we've seen. And so, you can have a nice big ultrasound image. But if it has to be the size of the anatomy, especially if you're a little presbyopic, like some of us are more than others, but, you don't get the full benefit of ultrasound with that little display. It's mainly for localization. So the problem would be more, can you identify the right vein? But if you can see it, you can hit it, basically, yeah.
Okay. I'd like to thank all the speakers for this afternoon and for the whole meeting, and thanks to everyone for staying until the end here. I know there's always a little bit of difficulty with people's flights and things, but thanks very much, and I hope everyone enjoyed their time. Thanks again.